So what we previously discussed is glycolysis, shown here in brief, going to the production of pyruvate. And we know that the glycolytic pathway has two parts, an energy-requiring phase, where we need ATP, and an energy-producing phase, where NAD+ is required to produce NADH. So anaerobic glycolysis occurs for a couple reasons. One is in the absence of oxygen, the electron transport chain can no longer oxidize our produced NADH to regenerate our NAD+. So under those conditions, we use our reaction by lactate dehydrogenase to convert pyruvate to lactate as a means of regenerating NAD+, as opposed to the electron transport chain. The other reason why anaerobic glycolysis may be occurring would be a block, as shown here, in the TCA cycle. So a deficiency in downstream enzymes to further oxidize pyruvate to carbon dioxide and water, as is normally done in the TCA cycle, would also lead to an elevation in pyruvate, which would increase production of lactate. So the enzyme responsible for anaerobic metabolism of pyruvate is shown here, and that's lactate dehydrogenase. So pyruvate, it can be reduced to lactate through the use of NADH in the cytosol. And again, the primary role of this is to regenerate NAD+ for the upstream reactions of glycolysis. So the Cori cycle is a process by which lactate produced in peripheral tissues can be transported to the liver and used in alternative pathways. Illustrated here is exercising skeletal muscle, and what we'll see in a couple weeks is that exercising skeletal muscle under anaerobic conditions will use skeletal muscle glycogen as a fuel source, and this glycogen will be converted into glucose 6-phosphate and will enter glycolysis. Under anaerobic conditions, this pyruvate generated is reduced to lactate, and the lactate is then transported through the bloodstream to the liver. This can then be reconverted to pyruvate using lactate dehydrogenase, which is reversible, and the pyruvate will enter gluconeogenesis to produce glucose, which can then be taken up by the skeletal muscle. The Cori cycle is also important in red blood cells as they lack mitochondria. Therefore, in the absence of mitochondria, in order to maintain glycolysis in the red blood cell, pyruvate is converted to lactate. So if we go back to a clinical correlate I used as an example earlier on, this will make a lot more sense now. And that was a discussion of MELAS, or mitochondrial encephalopathy, lactic acidosis, and stroke. And this was caused by a deficiency in complex 1 or 4 in the electron transport chain. So in this case, we ask the question is, how does this lead to lactic acidosis? And what we've just seen is that in the case of glycolysis, typically our pyruvate would go to the TCA cycle, and those intermediates would then be oxidized in the electron transport chain. So we have glucose going to pyruvate, pyruvate normally entering into the mitochondria, oxidized into the TCA cycle, and our reducing equivalents oxidized to ATP, and our reducing equivalents oxidized to ATP. However, in this case, a deficiency in the electron transport chain here will inhibit the regeneration of NAD+ through the electron transport chain, and subsequently, anaerobic metabolism will occur as a means of regenerating NAD+ using the enzyme lactate dehydrogenase.